It's so nice to have you here. Before we begin our sleep stories, I would really like to actively calm down with you. And so, let's breathe. Inhale for 5 seconds, breathing down and into your gut. Hold the breath for 7 seconds. Now breathe out for 10 seconds, noticing your body relaxing naturally. Now that we have tapped into our parasympathetic nervous system, responsible for lowering our heart rate, we can begin our sleep story. Sweetest dreams, you beautiful beings. All living things on this planet, animals, plants, even bacteria, are built up of a building block called the cell. The size of an animal cell is about 10 to 20 micrometers in diameter. Micrometers can also be known as microns. To give you something to scale that to, a human hair, on average, is about 70 microns thick. So looking at the bottom of one of your hair strands, a single cell is roughly 7 times smaller. To create a human, trillions of these cells work together as a well-curated, master-engineered machine. The instructions and the design plans for which are stored right within every cell, in the DNA. The cell is designed to protect the vital structures within it. A beautiful play of chemistry. The cell membrane is constructed with phospholipids, proteins, and other attached molecules, such as carbohydrates. The phospholipids being the key player in the cell membrane. They form a bilayer structure. Imagine a phospholipid like a balloon with 2 strings attached. A phosphate and glycerol head and 2 fatty acid chains made of unsaturated and saturated fatty acids. The structure of this phospholipid means that the phospholipid head is hydrophobic, or known as water-hating, because hydrophobic molecules aren't soluble in water. And in contrast, the fatty acid tails are hydrophilic, meaning that these are water-loving, and they are soluble in water. The balloon structures are organized with the 2 tails in the center of the bilayer, and the heads are facing outwards. One wall of the heads facing the outside of the cell, and one wall of heads facing the inside of the cell. Earlier I had mentioned that there can be attached molecules sometimes on this phospholipid membrane. The molecules that attach to the outside of the membrane, that's a key note. These molecules only ever attach to the outside, and that's because of the function that they play. These molecules attach to allow different sites of the membrane to have a different chemistry, and therefore behave differently. So sometimes, if a particular protein or another molecule of interest needs to bind to the cell to potentially deliver something to the cell and potentially instruct the cell to behave differently. That's a very interesting topic, and I'll speak more about it later. But for now, I want to tell you about the inside of the cell. Within the cell is a suspension, a liquid known as the cell cytoplasm. Contained within is water, ions, proteins, and other floating structures. The most notable floating structure is the nucleus. Within this is the DNA, the most important thing to the survival of all species that have ever lived. The DNA is the thing that the cell wants to protect the most. So understandably, the nucleus is surrounded by a membrane of its own. Usually, visually, the DNA is not how you would normally think it to be. When you imagine the DNA, you might imagine it to be in the form of chromosomes, which unless a cell is undergoing mitosis being cell division, then the DNA is normally stored in its form called chromatin. If you were to look at this visually, it would almost just look like a bunch of spaghetti in a bowl. There's two different types of chromatin, euchromatin and heterochromatin, one being a thicker bundle and the other being a thinner bundle of DNA. However, when it is time for the cell to undergo cell division, or mitosis, then the DNA begins to form into chromosomes, ready to be split. Along with how the different sites on the cell membrane work, we will also talk about mitosis at a later stage. However, let's continue with the inside of the cell. Outside of the nucleus is a very important structure called the endoplasmic reticulum. There are two types of endoplasmic reticulum. One is the rough endoplasmic reticulum, and the other is the smooth endoplasmic reticulum. The rough endoplasmic reticulum can be remembered distinctly by having bumps, like little rocks covering the outside of it. These little rock-like structures are ribosomes, ribosomes being essential for protein synthesis. And so the endoplasmic reticulum you can remember as being the protein machine. And in contrast, the smooth endoplasmic reticulum has no rock-like structures, no ribosomes on the outside of it. And the smooth endoplasmic reticulum can be remembered as being the lipid machine because it's responsible in the process of lipid synthesis. There is a structure within the cell known as the Golgi apparatus that's responsible for adding the finishing touches to things that the cell has made, for example, proteins or lipids. Within the Golgi apparatus, these proteins or lipids are carried within vesicles, which are like small sacs made up of cell membrane. Proteins, lipids, and many other things can be engulfed within these vesicles and can be transported from place to place, inside or outside of the cell. Within the Golgi apparatus, proteins or lipids, or anything else being made by the cell, will travel through the Golgi apparatus, being modified for whatever their final function will be. And once they are done within the Golgi apparatus, these vesicles will release whatever is within it, either inside of the cell, if it's needed there, or outside of the cell, if that's where it's needed. Going back into the cell, although there are many structures that are very notable and undeniably important, the last one that I will talk about is the mitochondria. Many of you will know the mitochondria as the powerhouse of the cell, and indeed it is, for this is where most of the steps of cellular respiration occur, cellular respiration being the process of energy generation, and therefore it is the powerhouse of the cell. The structure of the mitochondria itself you can imagine almost like a boat. And within this boat are large folds. And within these large folds are where most of the chemical reactions go down. Going to the outside of the cell, we go back to the presence of carbohydrates and proteins in the cellular membrane. Carbohydrates can be present on the cellular membrane and are always attached to either a protein or a lipid, therefore being known as a glycoprotein or a glycolipid. The presence of these carbohydrates can help with cellular adhesion and also work as a signaling molecule to help other molecules to find the place they need to go. Carbohydrates are found most of the time bound to proteins, and therefore generally you will find more glycoproteins than you will glycolipids on a cell. It's important to note, whatever you find on the outside of the cell will be completely determined by what kind of cell it is and how healthy the cell is as well. For example, if a cell is infected or is no longer fit for function, the cell may produce proteins that signal it for destruction. Other cells within your body will come and destroy the no longer functioning cells. The purpose of this is to make sure that all of the cells that are functioning within your body are healthy, and the DNA is healthy, most importantly. This is to avoid any mutations in the DNA from being replicated and potentially resulting in a tumor. The cell is quite extraordinarily equipped for dealing with any mutations or errors that might occur. An example of this is in mitosis, the process of somatic cell replication, a somatic cell being any cell apart from the sperm and the egg. For the replication of these types of cells is called meiosis. The cells are so meticulous at ensuring whatever they are replicating is healthy and functional. And so, before mitosis even occurs, there is a large interphase with 3 stages that are focused on ensuring the contents of the cell are healthy and will not cause damage to the functioning organism. There are multiple proteins that work to check the cells and make sure that they are healthy. And there are multiple requirements that a cell must meet to be deemed as acceptable for replication. For example, in the middle phase of interphase, called the S phase, where DNA replication occurs, the DNA will be checked and assessed for its quality. And if it does not meet the quality quota, then the cell may not be replicated. Additional to this, there are 2 other checkpoints, the G1 phase and the G2 phase. If the cell is able to get past all of these checkpoints, then it's deemed healthy and ready to be replicated. Your DNA has already been formed into chromosomes and doubled and is ready to be split. In 6 phases: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis, the cell is split. Cytokinesis is the final stage of mitosis, and in this stage, the cell has completely split its contents. Now there are 2 cells from 1, and the process continues on and on. When a cell is too old to function and is no longer healthy, it is then destroyed. and healthy cells continue to propagate. Such a short and brief description of the cell. The cell is so complex and is so important that I hope that this gave you just a small amount of love and appreciation for what's going on within you all the time. These little cells are working so hard to protect your DNA and to continue to replicate as healthy as possible, keeping you the best functioning version of yourself that you can be.